Imagine reading the entire instruction manual of a human being. This manual contains all the information that makes us who we are. Whole genome sequencing, or WGS, is the key to unlocking its secrets. It has opened up unprecedented opportunities in medicine and forensics. By understanding our genetic makeup, we can diagnose diseases earlier and develop personalized treatments. Whole genome sequencing, as the name suggests, is a comprehensive method of analyzing an organism's complete DNA sequence. Unlike other genetic tests that focus on specific genes or regions of the genome, WGS provides a complete picture of an individual's genetic makeup. This comprehensive approach is crucial for understanding the complex interplay of genes and their role in health and disease. WGS involves determining the order of the 4 chemical building blocks or bases that make up DNA. These bases, adenine, thymine, guanine, and cytosine, form a unique sequence that dictates the genetic instructions for an organism. By comparing the sequenced genome to a reference genome, scientists can identify variations that may be associated with specific traits or diseases. These variations can range from single base changes to larger insertions or deletions of DNA segments. DNA, or deoxyribonucleic acid, is the molecule that carries the genetic instructions for all living organisms. It is found in the nucleus of every cell and is organized into structures called chromosomes. Humans have 23 pairs of chromosomes, inheriting one set from each parent. DNA has a double helix structure resembling a twisted ladder. The sides of the ladder are made up of sugar and phosphate molecules, while the rungs are formed by pairs of nitrogenous bases. These bases, adenine, thymine, guanine, and cytosine, always pair up in a specific way, A with T and G with C. This specific base pairing is essential for DNA replication and protein synthesis. The sequence of these bases determines the genetic code, which dictates the production of proteins, the building blocks of life. Section 4. Decoding the genome. Sequencing technologies. Over the years, DNA sequencing technologies have advanced rapidly, making it faster, cheaper, and more accessible to sequence genomes. The first human genome sequencing project, completed in 2000, took over a decade and cost billions of dollars. Today, whole genome sequencing can be performed in a matter of days or even hours at a fraction of the cost. Early sequencing methods, like Sanger sequencing, were time-consuming and labor-intensive. However, the advent of next-generation sequencing, or NGS, technologies revolutionized the field of genomics. NGS platforms allow for massively parallel sequencing, meaning millions or even billions of DNA fragments can be sequenced simultaneously. These technological advancements have paved the way for large-scale genomic studies, enabling researchers to study the genetic basis of diseases in greater detail and develop personalized medicine approaches. Section 5. The journey of a genome, from sample to sequence. The process of whole genome sequencing begins with collecting a biological sample. This sample can be blood, saliva, tissue, or even hair follicles. The type of sample required depends on the specific research question or clinical application. Once the sample is collected, DNA is extracted and purified. This involves breaking open the cells and separating the DNA from other cellular components. The purified DNA is then fragmented into smaller pieces, as sequencing technologies work best with shorter DNA fragments. These DNA fragments are then sequenced using sophisticated machines. The sequencing process generates raw data in the form of DNA sequences. These raw sequences are then aligned and assembled to create a complete genome sequence. Section 6. The power of whole genome sequencing. Applications and future directions. Whole genome sequencing impacts medicine, biology, and forensics. In medicine, it transforms diagnosis, treatment, and prevention. Identifying genetic mutations helps develop personalized treatments. It aids in drug discovery and development. Understanding genetics leads to effective therapies. In biology, it studies evolution and disease spread. The future of whole genome sequencing is bright. As costs decrease, it will become routine in healthcare. From personalized medicine to disease surveillance, it revolutionizes health management.